Welcome, dear students. Today I am going to deal with environmental sensitive genetic male sterility. You see, by its name, you can understand that this is a type of genetic male sterility. It means the nuclear MS gene, which is responsible for creating male sterility in plants, its expression is controlled by the environment. Then the situation becomes environmental sensitive genetic male sterility. And particularly it has been observed in the environment. If you see, it is two types. EGMS, environmental sensitive genetic male sterility. It is of two types, PGMS and TGMS. When that male sterile gene is being influenced by the photoperiod, then that EGMS is known as photoperiod sensitive genetic male sterility. The next one, TGMS, when the male sterile gene is influenced by the temperature, then it is said to be thermosensitive genetic male sterility. Now we will be discussing one by one both the types, how it is behaving, how can it be utilized for your breeding program. So putting the example of rice, if you see in thermosensitive genetic male sterility, if we talk, the example rice, it has been observed in many rice varieties that once this gene is available, then it has been observed that critical temperature ranges from 23 degrees Celsius to 29 degrees Celsius in different male sterile gene. If you are putting the same gene in one variety, the critical temperature is different. If you are putting the same genotype, same MS gene, TGMS gene in another genotype, the critical temperature may differ. Maybe the different genes also, TGMS genes are also controlling, are influenced by the temperature, different temperature ranges. So it is called the temperature critical point of the MS gene to express. So you have to, when you are introducing the male sterile gene, TGMS gene in your material, you have to observe it, how it is behaving, how your variety is behaving, what is its critical temperature, critical point for expressing the MS gene in your variety in which you have introduced it. So I'm showing the example of rice. If you see in a Chinese TGMS line, which is known as PAI64S. PAI64S has critical limit of 23.3 degrees Celsius. It has been observed. When that gene, TGMS gene, has been introgressed into PAI64, it has become 64S. So has critical limit of 23.3 degrees Celsius. Once the temperature is going beyond 23.3 degrees Celsius, this allows the MS gene, male sterile gene, to express. When the temperature is below to that critical point, 23.3 degrees Celsius in PAI64S lines, then plants becomes normal. They behave male fertile, like normal plants. So it means for this particular male sterile line, you have to provide more than 23.3 degrees Celsius for expression of male sterility. In this way, TGMS is behaving. So the period of developmental sensitive to temperature lasts from the formation of pollen mother cells to meiosis in rice. Means, what is the practical use of this sentence? That for expression of MS gene in rice, in TGMS lines, you have to provide that temperature 23.3 degrees Celsius. Of course, just before start of flowering, and for consecutive 30 days you have to provide during flowering time. So the practical use of this sentence is that you have to provide during this period, this temperature during this period, this whole flowering time. So here if you see, the TGMS is governed by at least two recessive nuclear genes designated as TMS1 and TMS2 because it is recessive here in case of TGMS. So this is the behavior of TGMS line, and once you are using this EGMS line for developing the hybrids by using these lines, so those hybrids are called as two-line hybrids. And once you are producing the hybrid seeds through this system, this hybrid seed production is known as two-line system of hybrid seed production, whether you are using through PGMS or through TGMS. So one by one I will be dealing. So this is all about TGMS. I will be coming in next video about PGMS. Thanking you very much.